Om Namah Shivaya. Om Amriteshwari Namaha. Hello children. Today we are going to study cell, the structure and functioning unit of life. This is Seema Gupta from Amrita vidyalayam Punnol, Thalassery. And here this is a chapter belonging to 11th grade. So we are going to study this chapter in detail today. Now, when we see the introduction of this chapter, we find that Robert Hooke in 1665 was the first person to identify and see these cells in the bark of the tree. Later, many people they studied this cell in detail and they found that not only in the plants, in a lot of microorganisms, in higher organisms, we find different size and shape of the cell. And in this way, we say the biggest cell is the ostrich egg. Now, in our body only we find different size and shape of the cell, like our RBCs, nerve cells, our muscular cells. So we see different shape of the cell. Now, according to this, in this nature, we have divided the cells into two different categories. One is called as a prokaryotic cell, the other one is a eukaryotic cell. The major difference between this eukaryotic and prokaryotic cell is the prokaryotic cell do not have an instinct nucleus. They don't have a nucleus and their genetic material is not been preserved inside that nucleus. As in the eukaryotic organisms, we find distinct nucleus. Further, let us see more about this prokaryotic cell. So here we study more about the bacteria. One of the other type of prokaryotic cell is the mycoplasmas or the archaebacteria. So now we see the bacteria in the nature in different shapes. Spherical, rod, spiral and vibro are the different shapes of bacteria we find in this nature. Now when we see in the bacteria in detail, we find that externally the bacteria is having a different coatings or the structures which cover or protect the bacteria, and internally it is having the cytoplasm. The DNA material is freely suspended in the cytoplasm, and externally it shows three layers of covering. The external is called as the Glycocalyx, the middle one is called cell wall, and the innermost is called as the plasma membrane. Now inside the cytoplasm, we find that the DNA is suspended freely inside the cytoplasm without any nucleus, and other than the genetical DNA, we find spherical DNA is called as plasmids, which is found in this cytoplasm. Further, when we see the modifications of this different layers around the bacteria, we find first modification in Glycocalyx. In Glycocalyx, we find two types. One, some of the bacteria will be having slime and thin and flexible layer of Glycocalyx. Some other type of bacteria will be having tough and thick coat of Glycocalyx. And according to this modification, when we put the gram stain, we can differentiate these bacteria into two types, gram positive bacteria and gram negative bacteria. So here we can see all the blue colors are the gram positive bacteria, the pink colors are gram negative bacteria. The second modification we can see in the plasma membrane, where we can see the internal folding inside the plasma membrane, which is called as mesosomes, which are very important for cell-to-cell communication, for transportation of different material. In our bacteria, we can see the bacteria locomotes, or the locomotion in the bacteria is by the three things, is by the three structures. One is the fimbriae, second one is flagellum, and third one is the pilus. Now if we see the structure of flagellum in detail, we see that the flagellum is attached internally to the plasma membrane, where we call it as basal body. Further, it is attached to the cell wall, from where we call it as hook, and from the hook it is a single layer extension of plasma membrane, which we call it as filament. Further, other than this all, we find ribosomes, which play very important role in bacteria for the production of different enzymes in them. Now these ribosomes in the bacteria are of 70S or 70 subunit. Now the 70S, we also find it as 50 and 30S when they are non-functional. Now the last topic in this is inclusion bodies in bacteria. Whenever the bacteria want to store something which is extra enzymes or some extra food or some waste products, everything it stores for temporary basis in this inclusion bodies, which in this diagram we can see as the black dots. They are the small vesicle like things which is present inside the cytoplasm. Thank you so much, children. I hope you understood all the topics. Hope you have a very good day. Thank you so much.